Good evening, everyone. Welcome back to the second session, another highly informative session, this time about vaccination and treatment. And in addition, at the end of the first two presentations, there'll be a panel discussion, and Dominic Dwyer, a virologist who's currently in Wuhan with a WHO group looking at the origins of coronavirus, will also join us. So our first speaker is Professor Paul Griffin. Paul's an ID physician at Mater Health in Brisbane and also a researcher with QMIR. And Paul's going to talk to us, give an overview of vaccines. Thanks, Paul.
Well, thanks very much for the invitation to speak. I feel very privileged, particularly given the caliber of presenters we've already seen and about to see. And, obviously it would have been nice to be there in person, but it's great that we could hold this meeting virtually. And I just want to thank the organisers, Kim Sampson and the Immunisation Coalition, for having a meeting that I guess couldn't be more topical right now. So I'm going to very quickly, in 25 minutes that I've been allocated, try and run everybody through the vaccines. It's a lot to cover and there's a bit of repetition, but I've just tried to group things in certain ways to hopefully make some key points a little bit clearer. I do have some relevant disclosures. I'm trialling 4 vaccines at the moment. I've approved some of these in some of my other roles and am on the International Medical Advisory Board for AstraZeneca. So here's a quick outline and I'll get straight into things. So, obviously this is a very learned audience with a lot of interest in this space. So I won't go through the background too much, just to say that we did have a lot of promising candidates for sales upon which a lot of the current vaccines have been built. Well, we did have some complications there that fortunately we haven't seen with these COVID-19 vaccines. And obviously a lot of the limitations on the clinical trials for the first SARS vaccines was because we actually eradicated that disease before the later phase trials could happen. Not that there weren't promising candidates that could have potentially been taken forward had we had the need. Just wanted to quickly touch on some objectives that we are looking at in some of these clinical trials, because obviously there's never been more discussion in the mainstream media about clinical trials and outcomes and endpoints and all that sort of thing. And just to say that, the primary outcome in the phase 3 trials, those numbers we're hearing so much about in terms of the efficacy of these vaccines, is prevention of symptomatic microbiologically or PCR proven disease. So while that's obviously a really telling number and really important and gives us a good indication of efficacy, of course, there's a lot more to that. We're obviously still monitoring the safety, tolerability and reactogenicity, but there's so many more other endpoints that are being measured as secondary objectives, mostly because the numbers are hard to achieve to reach significance. The laboratory correlates of protection are still monitored, and a lot more detailed clinical responses that we're not hearing about in the preprints or the press releases just yet, although we did obviously hear the asymptomatic infection rate from Novavax the other day, and that was promising. Phase 2 is still mostly focused on the laboratory correlates, and still obviously safety. And we do look at some of those things, but obviously very underpowered for that. And we're hearing a lot of reports of these vaccine studies where they're merging all 3 of these, including the phase 1, where really that's about safety, tolerability and reactogenicity. So we don't see a lot of cases and obviously very small numbers there. I couldn't do a vaccine talk without just touching on some of the issues we've faced. And obviously it's been great to see the progress happen as fast as it has, but I think that's lent itself to people being a little bit skeptic, or a little bit of skepticism about how we're able to do that. And I just wanted to touch on some of those points. Obviously funding was the key driver there. A lot of big organizations like CEPI and also governments gave a lot of funding for these vaccine candidates. And that then obviously led to a whole host of things, including the scaling of manufacturing, which has been a huge part of how we've been able to do this so quickly. In terms of preclinical data, we still needed the toxicity data to commence our human trials, but we often were able to commence the submissions before those final reports were done. And the efficacy data we could often do in parallel with the safety assessments in humans. So what that really means is animal challenge studies were actually happening in parallel to the phase 1 studies in humans. We had a lot of support from our regulatory bodies, and I've got a little graph there that shows how quickly we were able to get some of our studies up and running from first submission to commence dosing. And it was often around 40 to 50 days, which might sound like a lot, but realistically in the clinical trial space, that's essentially unheard of. Study design, we often combine phase 1 and 2 studies. So we still needed to obviously see the endpoints from the phase 1 study that allowed us to progress. We didn't have to conclude that study and go through a regulatory process with the phase 2. The tracker, I think everybody in this audience is probably aware of where these are up to. Obviously, there are hundreds of candidates, and these are the ones that are progressing through clinical trials at the moment. And many are actually in every stage of clinical trials simultaneously. So it's hard to know exactly how many are out there. But as I say, there are hundreds of really promising candidates at the moment. Just thought I'd quickly go through the main mechanisms that we're using here. Obviously, the nucleic acid mechanism is the one we're hearing about a lot, because that includes Pfizer, which has a 95% efficacy and has been approved. Moderna, very similar technology, similar efficacy, also approved. But there are a lot of others that are using this same technology. There are quite a few other mRNAs, and I've listed a few there, and a lot of DNA vaccines, which in this country, at least, we're not hearing quite as much about. But I did want to point out, and while I've focused on the ones that are in later phase trials in those lists, that there is one at the bottom there, Simvivo, that I've just started the phase 1 trial here in Brisbane, and that's an oral vaccine. I'll just touch on that later because it's very interesting technology. So in terms of the mechanism of action, so obviously mRNA is the instruction to make proteins. And so the cellular machinery inside the recipient, the ribosome, translates these into proteins. And in this case, obviously, we're talking about the spike. But one of the downfalls of mRNA is it's inherently unstable. That's good in some ways, but obviously a challenge when we're trying to use that therapeutically or for vaccine development. So it needs to be coated in something to prevent degradation and needs to be kept very cold. And I'm sure everyone's familiar with the extreme cold chain logistical challenges we're having with some of those at the moment. Fortunately, the mRNA itself helps to boost the immune response, so we often don't need adjuvants, but do need multiple doses. And again, that's what we've seen with those lead candidates. It's fast. Again, we've seen those be the first to be developed and approved. Not previously licensed, but very safe technology. Viral vector, again, we're hearing a lot about some of these, particularly AstraZeneca, and that top one, which gets called a lot of things, but I like Sputnik 5. I think that's a cool name. But again, there's a lot of others in that group as well, including the Johnson & Johnson vaccine. I've listed the efficacy rates there and the approvals, but I'll touch on those in a slightly different format a little bit later. And how this works, instead of administering the instructions for those proteins, we separately in mRNA form, we actually put those into a vector, so a benign virus, and commonly it's adenoviruses in the majority of the COVID vaccines we're hearing about. AstraZeneca, it's a chimpanzee adenovirus, and others it's human benign adenovirus. So this virus can enter the cells but doesn't replicate. Some can, though. There are some replicating ones, but again, we haven't heard about those quite as commonly. So that the instructions for the spike protein translate all the way through to proteins, and we can then generate an immune response against those spike proteins. And the vector virus also helps in terms of the generation of the immune response. So we don't actually need an adjuvant for these, but we also usually get very potent cellular as well as humoral responses, so a broad immune response, which is good. But some immunity, whether it be pre-existing or generated from receipt of the vaccine or the previous dose of the vaccine, may impact on efficacy. And we've seen various mechanisms, including using a different vector for the first and subsequent dose as a way of addressing that potentially. Protein vaccines, we've heard about a lot of these. Novavax is probably the front runner there that a lot of us have heard about, but there's quite a few in development. The Clover vaccine was also trialled in Australia, and I'll just touch on that. And the way they work is we actually make that target antigen in the laboratory, but often it has some problems in terms of either forming aggregates or folding into such a way that it won't give the immune response against the native antigen. So we now these days typically combine it with some clever technology. In the case of Novavax, for example, it's nanoparticles. And I think most people would have heard about the University of Queensland's molecular clamp approach. These are fairly easy to manufacture, so we obviously don't need cell culture or living virus to make these. We can do it essentially from the sequence, and it's a well-established platform. Can be a bit costly, and often we need to do some things to help the immune response along, including, for example, adding adjuvants or multiple doses. In terms of inactivated or attenuated vaccines, obviously a tried and tested approach, been around for a long time, that we haven't heard quite so much about just yet in terms of COVID. There's a few listed there. I think some other parts of the world are a little bit more involved in this than what we've heard about. And there is an interesting one from Codagenics, which is a live attenuated intranasal vaccine, which uses their proprietary de-optimization technology, which basically introduces these silent mutations. So it looks really similar, but is a very benign live attenuated virus. And I think we'll hear more about theirs as they progress through. So how that works, we create weakened or killed viruses. A lot of them are using a strain of virus that's able to multiply quickly in cell culture in monkey kidney cells. And that's been attenuated using this chemical beta-propiolactone, which binds to the genes and means that the virus can't replicate. But all the proteins, particularly on the surface, including spike, for example, remain intact. So these are fairly easy to scale and relatively inexpensive, and obviously an approach we've used for a very long time. But often we still need something to help boost the immune response, such as an adjuvant. And so far, at least, we haven't seen the same levels of efficacy we've seen, for example, with the mRNA approach. So I just thought I'd quickly touch on a few of the lead vaccines now. And again, a lot of this information will be familiar with most people in the audience, but I just thought I'd quickly summarize it in this form. So the Pfizer-BioNTech vaccine, the first one to be approved here, you can see there is still in phase 2 as well as phase 3 trials and has been approved in several countries. Intramuscular, 2 doses 3 weeks apart, and has that cold chain issue. And just some evidence there. So the NEJM paper was at the end of December and showed that efficacy rate of 95%. And we've seen some recent preprints that suggest that it's still likely to have a fairly reasonable effect against the new variants, which is obviously the latest challenge we're facing. So Moderna, the other mRNA vaccine, again, we had a fairly recent NEJM paper from the 4th of February, very large study, again, that showed that really good efficacy rate that's been quoted at 94.1 or 94.5%, and some recent press releases as well suggesting that one too might have some reduction in neutralizing antibody titers, but still likely to be effective. And in terms of evidence, I did just want to point out with that little article at the bottom that while everyone's saying this is brand new technology, we have been looking at that for a while. I was involved in a study a few years ago looking at an mRNA-based RSV vaccine, and that was just recently published. So while it's kind of the flavor of the month because it seems to be very effective here, we have been looking at that for a little while. Oxford AstraZeneca, again, I think people are familiar with this. This is one of the other vaccines that we have a purchasing agreement for in this country. And efficacy, well, that's a bit of a challenging one and has kind of evolved over time. We saw these two really good papers in the Lancet, and they published some fairly early results. I think that was July, that first Lancet paper, fairly small study, the phase 1 and 2, showed it looks pretty good, but you did need that homologous boosting, so the second dose. And then the second study in November showed a few things, similar immunogenicity, but maybe better tolerated in the old than the young. And I guess that's where we get to the next paper, that I think was the 8th of December, that caused all the controversy in terms of variable efficacy rates, depending on a few factors, with the intended dosing perhaps a little bit underwhelming at 62%, but the pooled analysis in that study demonstrated 70% efficacy. But fortunately, we've had some more information come through, a preprint in the Lancet from just last week that suggested with an increased dosing interval of more than 12 weeks, perhaps the efficacy of that vaccine may well be 82.4%. So again, a very promising vaccine with a good evidence base, with some more questions to be answered with further evidence to come. J&J is an interesting candidate, given its single dose, and some variable efficacy reported in those studies. We've seen one NEJM study from January this year, which again showed some promise, and then a recent press release, so not yet published, but this is the efficacy data that most people are quoting for that vaccine now. Excuse me, 72% in the US, but perhaps lower elsewhere, but still good at preventing severe disease. And so obviously we'll wait to see the peer-reviewed full publication there. Again, another promising vaccine with a good emerging evidence base. And Novavax, and this was one of the studies that I was part of, published the phase 1/2 a little while ago, which did show some good promising early data, and a press release from just over 2 weeks ago, 89% from their UK phase 3 study. So it looks to be a promising vaccine as well. And the team at Novavax shared this slide with me just this week, again, showing a few things, but the overall efficacy of 89%. The original strain maybe a bit more, the UK variant maybe a little bit less, but still very promising efficacy. It was interesting in the South African parts of their study, prior protection didn't seem to protect against the new variant, but their vaccine did. So, and they're also, the final point there is they're making some new vaccines to address those variants as well. So I just thought I'd do some comparisons because we're hearing so much about these vaccines, and I thought it'd be useful to maybe draw together some comparisons here. So you can see in terms of efficacy, it's the Pfizer-BioNTech that wins there, but both the mRNAs are fairly close. Janssen is probably a little bit behind. In terms of protection from severe disease, and with the debate about whether these vaccines have transmission blocking capacity, you can see there, they're all very good at preventing severe disease. So even that in itself, I think, is just amazing. And so we should all be very, very excited about these vaccines being rolled out. You can see the majority are 2 doses with varying intervals. Oxford, it might be 12 weeks plus based on some of that newer data. The Janssen J&J candidate is 1 dose, which is interesting. All similar in terms of age, although the Pfizer-BioNTech is saying 16 plus. And then in terms of storage, I just thought I'd put there, this is incredibly complicated if anyone's having a good look at this. Ultra cold, -70 for 6 months, and then normal refrigeration temperatures for 5 days. Moderna is still called ultra cold, but -20 for 6 months, and then refrigerated for up to 30 days, and apparently room temperature stable for up to 12 hours. And the others stable at essentially refrigerated temperatures. And so I'll just come back, that primary efficacy, as I say, that endpoint is that microbiologically proven symptomatic infection. In terms of some other comparisons, so I've got those efficacy rates there at the top. The UK variant, again, waiting for some really robust data here, although it's beginning to emerge. Thought that it's not going to have a big factor there. Novavax say that slight reduction, but still approaching 90% with those new variants. In terms of the South African variant, some work to suggest there's a decrease in antibodies, but this doesn't correlate with efficacy. They think those reduced neutralizing antibodies will still be sufficient. The J&J candidate was reported as 57%, which again may seem to be low, but again still very promising. And Novavax have released some information there that in their all comers, a bit less than 50, but HIV negative was a little bit higher. And in terms of contingency for new variants, Pfizer not yet developing a booster. Moderna are, Oxford AstraZeneca are developing an updated vaccine, as are Novavax. So I just thought I'd quickly touch on Australia's contribution as well. So Novavax was commenced here in Australia. I started that phase 1 trial on the 26th of May. As said, the recombinant spike protein with that matrix M adjuvant. The phase 1 for that study was published in the New England, as I said before. Went on to a phase 2 in Australia, as well as a lot of other countries, and the phase 3 commenced in September 24. And this is one of the 4, now 3 vaccines with which Australia has an agreement for at 51 million doses. And while not being talked about quite as much as the other candidates, I think this will be a really important part of our strategy. The University of Queensland vaccine, and we could have a whole session talking about that. That was the recombinant spike protein with the molecular clamp. First dose up here in Brisbane on the 13th of July. And this arose from a rapid response project that was commenced around 2 years ago. So it had been in the works for some time with some very clever people in the lab, Keith Chappell, Paul Young, and CEPI provided some generous funding to that project well before COVID was a thing. The phase 1 was still going through with that, and it still did look like it had excellent preclinical data, and the publication should be out soon. But we did run into that diagnostic interference issue, which unfortunately was the undoing of that candidate. Some others, the Clover vaccine and S-Trimer from a Chinese-based company was undertaken in Perth. We've heard about vaccine at Flinders University in South Australia, some promising reports in the media there. And we've just started a trial also with the Serum Institute of India with an RBD, interesting technology formed into a virus-like protein. And there's plans to certainly scale that up and proceed with further clinical trials of that one. I just wanted to touch on the other vaccine trial that we've just started, which is really interesting because it's an oral vaccine. And as far as we're aware, the first oral vaccine in clinical trials. There are certainly intradermal, transdermal, and intranasal platforms well underway, but we believe this is the first oral. And it's a really clever technology with some genetically modified, basically probiotic bacteria that deliver the DNA encoding the spike protein. And it's thought that that'll be expressed in such a way that it will develop immunity with a mucosal element, which should hopefully be helpful. And that's very stable at room temperature as well. So if that proves efficacious in clinical trials, I think that will be exciting. In terms of the vaccines that are approved, orange here or yellow, depending on how it appears on your screen, are the ones that have some kind of emergency use approval, and green is actually approved. And Sinopharm, which is an inactivated vaccine, quoted efficacy of around 80, was approved quite early in China and has emergency use in a number of countries. Pfizer-BioNTech, which we're obviously on board with and approved now in Australia, is obviously approved in many countries, as is Moderna. Only, and I'm not sure if you can see, the green is only in Switzerland, but has emergency use authorization in a large number of countries. Sputnik 5 was approved for early use in Russia, but actually has emergency use in many other countries now. And Oxford AstraZeneca, again, given that's one that we'll be getting, I think a lot of people are familiar with this, emergency use in the UK, the EU, and by that orange there, you can see there's many countries now where that is approved. And CanSino, Chinese-based vaccine, has early approval in China as well. There are some other vaccines that I've mentioned that I've grouped in some of those leading vaccines, but not yet approved. But I think that's imminent, should happen almost any day, I guess, that the J&J candidate, as well as Novavax, with some interesting evidence emerging there. Unfortunately, there's a slide on abandoned vaccines, that the UQ candidate is not the only one that appears here. This was abandoned on December 10, the first vaccine, unfortunately, to be abandoned after entering clinical trials due to that HIV test interference. Merck have had 2 candidates they've been collaborating on that have been abandoned, largely due to the response being weaker than natural infection. The final one is a UK candidate that they thought they've got a pretty good vaccine already, so they thought they wouldn't embark on a new study just yet. In terms of our supply, and there's people that have already spoken, and I'm sure in the audience that know more about this than I, but I just thought I'd quickly run through this. We're in an excellent position, obviously. Pfizer-BioNTech, now 20 million doses, does have those challenging logistics, but it'll be great to get started with that fairly soon. Oxford AstraZeneca, we're getting the first small allocation delivered, but then CSL are going to be manufacturing a very large amount of that. So having manufacturing onshore is obviously a huge plus, given some of the issues we've been seeing. Novavax, we're scheduled to receive 51 million doses of that vaccine once the data is sufficient to approve it. I know a lot of people have been saying 150 million doses, but unless my maths is wrong or I've misunderstood there, I think that's about 125 million doses, which is obviously amazing for this country to be in such a good position. And I've included the UQ vaccine there because that would have been there had we not had that slight issue. So just to wrap all that up, I know it's a bit of a whirlwind tour through a lot of elements of those vaccines. I just wanted to put some questions that we're still asked really often. Is it true the vaccines won't tell us about prevention of mild disease, more severe disease, or transmission blocking? And no, that's not true. So as I mentioned a few times, that main endpoint about symptomatic microbiologically proven disease is the one that is easy to measure in trials, so we can get enough numbers to make that robust statistically. But we look at all sorts of other endpoints, and in the fullness of time, when they're analysed, we'll get a better idea of the full spectrum of protection. They all look really good at severe disease, but the virological assessment, even in asymptomatic patients, whilst not perfect, will be a useful surrogate as an early indication, I guess, of whether they will likely reduce shedding, which might equate to transmission blocking. It doesn't match up 100%, and a lot of people were critical of discussions to that effect around Novavax, but really useful information that we will see soon. And is it true the vaccines won't reduce transmission? I think they all will to some degree. It's anywhere between 0 and 100, and we don't know any more than that just yet. But I think what's clear is there will be an impact. And even if there isn't, the ability to reduce severe disease is going to be just amazing for us. There's a lot of outstanding questions that we will get more information over the coming weeks and months. Longevity and requirement for boosting. We will get information there, but it remains to be seen yet. Lots of talk about heterologous boosting and combining different vaccines. And the simple answer there is we can't say yet, but it looks like a really promising approach. And those studies will come. So we'll get an idea there. And that might boost how all of our vaccines work and I guess address some relative shortcomings of the current vaccines that we've been talking about. The biggest challenge, and I think everyone in this group would be aware of this and is here partly to try and address that, is uptake. It's no good having a vaccine that's highly efficacious, but not taken. And so, I think this is what our focus really needs to be. And obviously there's some great work happening at so many levels in this country at the moment. And fortunately, we're a country that traditionally does well in this space and has a lot of common sense. So I think we'll do relatively well, but it's something we need to keep working on. And so I think I'm just about at my allocated time. It's been great to see these vaccines develop faster than ever before without omitting any of the critical steps. So we're just as confident they're safe and efficacious as we would be normally with any vaccine. We've applied every existing platform and obviously many that didn't exist to this. We've got so many exceptional candidates and so many that are now approved, which is fantastic. We're still developing vaccines though, because we don't have a perfect vaccine and we probably won't. So we're going to need more vaccines to come through to hopefully increase how these vaccines perform. Scaling of manufacturing has put us in a great position, but we've still got some challenges to overcome. And, I guess given that and some of the issues around transmission blocking, it's just really important that everybody knows. And again, I'm sure everyone here is completely on board with this, that, the vaccine is amazing and will be a key intervention, but not the whole story. We still need testing, therapies, other ways of helping with contact tracing, for example. And of course, to keep up the good work with our other mitigation strategies. But hopefully we'll see those start to, within reason, trickle back as the vaccine coverage rolls out. And so that's where I'll leave it there. Thank you, everybody.
Paul, thank you. Absolutely masterful. And we're very lucky to have someone of your expertise and clear enthusiasm for vaccines in Australia. I'll introduce the next speaker, Professor Stephen Tong. Stephen Tong's a colleague of mine at Royal Melbourne Hospital. He's part of the Victorian Infectious Diseases Service. He's also a researcher at the Doherty Institute and is the lead investigator on ASCOT, which is an adaptive platform to trial new treatments for COVID. Steve, thanks for joining us. I look forward to hearing from you.
Thanks very much, Lou, and delighted to be part of this, and thanks for the invitation from the Immunisation Coalition. Before I start, I'd like to just acknowledge the traditional custodians of the lands upon which we meet and pay my respects to elders past, present and emerging, and also to say that I don't have any specific conflicts of interest apart from running the ASCOT trial. So early last year, in about March, there started to be some publications suggesting hydroxychloroquine was going to be an effective treatment. And this was probably the first of those publications from a group in France, where patients who received hydroxychloroquine compared to controls seemed to reduce the viral load of SARS-CoV-2 fairly rapidly in a very statistically significant manner. There are a number of further observational studies which found similar kind of findings. And based on all that, as you may know, Donald Trump suggests that hydroxychloroquine and azithromycin have a real chance to be the biggest game changer in the history of medicine, and that everyone should use these drugs immediately. And of course, many people did, unfortunately, and across the United States and South America and probably Europe as well, many patients were put onto hydroxychloroquine based on this observational data. Subsequently, there's been a number of randomized clinical trials, the largest being the recovery trial from the UK, and I'll speak multiple times about recovery, a UK-based randomized controlled trial looking at multiple different treatments, but in this case for hydroxychloroquine. And amongst over 4,700 patients, they showed that there was no difference in the mortality rate. So on these graphs, there's a mortality on the y-axis and time since starting the treatment on the x-axis. And over time, there really was no difference in mortality. If anything, hydroxychloroquine looked like it was going to be causing more mortality than the control groups. This was followed by meta-analyses of further randomized controlled trials, and most of these trials confirmed what the recovery study demonstrated, that there was no difference in mortality. And when you added up all these studies together, including this WHO Solidarity trial, which was across numerous countries, in fact, it looked like hydroxychloroquine was associated with increased mortality with an odds ratio of 1.12. And so not only was it not effective, but perhaps may have been associated with worse outcomes. So Professor Darryl Francis is a cardiologist and epidemiologist in Imperial College in the United Kingdom. And he wrote around this time, this is May the 2nd, that if we decide if treatments are beneficial by doing scientific experiments, that is randomized controlled trials. And there's some people in medical fields who would suggest that randomized controlled trials are actually medicine's greatest advance in this past century and a half, that doing randomized controlled trials has allowed us to work out what things work and what things don't. And he wrote that this doesn't mean he doesn't look at non-randomized data when he wants to know things. So if you want to know how many people, the proportion of people who are men, or what proportion of COVID patients get ventilated, but never to see if a treatment works or whether it's beneficial or not. And in fact, he makes the point that observational data is pointless when you want to find out whether a therapy works or not. I'm going to come back to all that, but just before I go on, there's been a bit of controversy recently in Australia about both hydroxychloroquine and ivermectin. And interestingly, Merck, which is a company that manufactures, produces and sells ivermectin, as of 3 days ago, made this press release. And if you think about it, if this is the company that's selling the drug, they have the most to gain for in promoting the use of this drug and selling it. But they clearly note that their analysis has identified no scientific basis for potential therapeutic effect against COVID-19 from preclinical studies, no meaningful evidence for clinical activity or efficacy in patients with COVID-19, and a concerning lack of safety data in the majority of studies. So basically, for both hydroxychloroquine and ivermectin, there's absolutely no evidence that they're beneficial at this stage. And I think we should put that message out very strongly, as opposed to those who are feeding us misinformation about these two medications. So the overview to the study, I've talked about the observational versus randomized data and the importance of randomized clinical trials. I'm going to talk a bit about disease stages, numbers as well, and then more specifically about the various therapeutics. So the disease stages. So COVID has got a very remarkable range of kind of manifestations, going from mild through to severe and death. Most patients obviously get mild COVID. And in that case, infection or exposure to the infection to SARS-CoV-2 starts at about day -5 prior to the symptom onset, which is at a median of about 5 days after infection. And at that time, there's a lot of viral replication. And then this dies down. So in mild cases, most of the time, by about day 7 or 8, most of the viral replication has finished. And then there's a growing immune response, growing antibody response, and then symptom relief and recovery. In contrast, in severe COVID, there is that ongoing viral replication early on that seems to last a bit longer, perhaps due to a less strong immune response in that person. But with the ongoing viral replication, there then develops this hyperinflammatory phase of infection, or really of the immune response, that leads to the complications of acute respiratory distress syndrome, patients needing to be ventilated, going to the intensive care unit, and for some of those patients ultimately dying. So quite different kind of, I guess, phases of illness, starting from this early viral replication through to an aberrant inflammatory response. And so the kinds of therapies that we might use might differ according to where we are along this time course. So from early pre-symptomatic to mild and moderate illness, viral replication looks like it's important. So things like antiviral therapies and antibody therapies should be effective. But then later on in illness, where inflammation really kicks in, we need to probably use anti-inflammatory medications rather than targeting the virus itself. But this does make it difficult. So the reality is that most people don't get particularly sick from COVID. If you had 200, there's 200 dots there representing 200 people who get the infection. Only about 11 of those will end up being hospitalized, so around 5%. And of those, 3 end up in the intensive care unit, and of those, 1 person dies. So you can imagine, as we look at this mild and severe spectrum of disease, often what we're trying to do is to find things that will stop people from getting to hospital, stop them from dying. But when we have to treat a large population, it's very difficult to predict which of these patients will actually end up being the one that dies. And so we can be giving treatments that may be quite expensive early on in disease, but in fact, the majority of those people were going to recover anyway and wouldn't benefit from that intervention. So it's been really tricky trying to work out that balance between giving medications that at an early stage that might be most effective at that time point, but the reality being that most of those people didn't actually need that medication. Whereas once we get out to more the severe phase, you're going to always get more bang for your buck in terms of reducing the number of people who might die with an effective therapy. So I'm going to move on to the specific therapies now. So antibody therapies are ones which target with antibodies that either the body has produced or that are produced artificially against the virus. And typically we're thinking about impacting upon the spike protein. So again, going back to this diagram, we think that these antibody therapies are most likely to be useful early on in the disease when it targets the viral replication. So convalescent plasma was probably the first one off the block. This is taking plasma from someone who's recovered and already generated antibodies and infusing that plasma to someone who's newly acutely unwell with COVID. In the United States, there was an expanded access program and over 100,000 people received convalescent plasma as part of this, and they needed to consent to this. Data was collected on these patients, but unfortunately, not one of those patients was randomized. They all just got the convalescent plasma. So this actually provided no data as to whether convalescent plasma worked or not. There has been some data to suggest that if the plasma was of high titer, so if there were more antibodies in it, and if it was given earlier in the disease course, that it was more effective than if it was low titer or if it was given later in the disease course. But we still had no idea whether it actually worked overall or not. And then this recovery trial again, there was a press release on January the 15th. Amongst 10,000 odd randomized patients, there was absolutely no difference in the 28-day mortality, so 18% versus 18%. And this agreed with other randomized controlled trials as well, which suggested that there was little benefit in convalescent plasma. So I think overall, we can say that there's no benefit in reducing mortality. Maybe a possible benefit if it's a very high titer convalescent plasma and if it's given very early on. The monoclonal antibodies, these are the designer antibodies, and there's been two companies which have been putting these into clinical trials principally, so Eli Lilly and Regeneron. Again, within hospitalized patients, there's been a couple of trials. So these are patients who are in hospital, presenting further down the illness, where it didn't seem to be of any benefit. However, there's been two outpatient trials. So these are patients who've been diagnosed within the last 3 days, within 3 days of their symptom onset, and giving these patients the monoclonal antibodies seem to reduce the number of patients who then subsequently either needed hospitalization or presentation to the emergency departments. So it looks like it's more effective if it's given earlier, before the endogenous, the person's antibody response has developed. Again, these are considered pretty high titers as well. The problem is, though, that it's logistically very difficult. So it's logistically difficult to get people in, one, to diagnose them quickly and early, then get them to a hospital center, which can provide an intravenous infusion. And to do this at scale is proving to be very difficult. And even then, at about $3,000 or $4,000 per infusion, it's an expensive way to go when the majority of these patients are unlikely to get particularly sick with their COVID infection. The next thing is antivirals. So the virus has to replicate, and there are various drugs that prevent or reduce the replication of the virus. And many of these drugs have been shown in cell culture and laboratory experiments to reduce that replication cycle. And some of them have also been used during the first SARS and observational data principally to show that they might be beneficial. So again, back to this diagram, we're targeting this viral replication with antiviral therapy. Ideally, you would kind of have some oral safe therapy that you could give very early on. And unfortunately, we don't have any of those at the moment. And most of these medications are being tested in those with really moderate illness mainly. So by the time they get into hospital. So I'm going back to the recovery trial because they've done an enormous job in looking at multiple different antiviral therapies. The first being hydroxychloroquine, and I've shown this slide before. Again, on the y-axis is the mortality, x-axis the time since randomization starting that treatment, and there was no difference in mortality with hydroxychloroquine compared to usual care amongst about, what was that, 4,700 patients or so. Lopinavir ritonavir, which is also called Kaletra, is an anti-HIV medication, seems to have some in vitro activity in cell culture. And again, absolutely no difference amongst, about 5,000 patients between lopinavir ritonavir and usual care. So this didn't work either. And finally, azithromycin, which is an antibacterial, also anti-inflammatory, and has some effect against SARS-CoV-2. Again, absolutely no difference in mortality or any other outcomes that they looked at. And I guess just to remember that all these drugs showed some activity in vitro, all had various, biological plausibility, and that's why they got into such a big trial for testing, and all of them were shown not to work, and really demonstrates that biological plausibility doesn't usually translate to clinical effectiveness. And you don't know that until you run the randomized controlled trial. The WHO Solidarity trial was across multiple countries and run by the WHO. They also looked at hydroxychloroquine. Again, no difference amongst 2,000 patients or so. Lopinavir ritonavir, again, no difference amongst, in this case, close to 2,600 patients. Interferon, which is meant to generate a better immune response against SARS-CoV-2, again, no difference. And if anything, interferon looked like it was being associated with increased mortality compared to control group. And finally, remdesivir, which is a little bit more controversial, but amongst 5,500 patients, there was no difference between the control group and remdesivir, a kind of more targeted antiviral in this WHO Solidarity trial. So I remember when this study came out, and it was really quite depressing in that all these studies, all these kind of agents that we were trying and hoping would work, none of them were shown to be of clinical effectiveness in reducing mortality. So the WHO has actually recommended against the use of remdesivir, which is different from what the United States and what Australia has actually recommended. So there was one other large trial run by the National Institutes of Health in the United States that compared remdesivir to placebo. And this one was a placebo comparative trial. In this case, the y-axis is the proportion of patients who recover from their illness. And you can see that remdesivir patients tended to recover at an earlier stage compared to those who received placebo. And that was particularly the case for patients who are hospitalized and receiving oxygen therapy. You can see that there's a good split between the two arms, whereas this was not the case for patients who did not need oxygen, and nor was it the case for patients who were on non-invasive mechanical ventilation or mechanical ventilation. The mortality rates were lower in remdesivir, although this was not statistically significant, so 11.4% versus 15.2%. But in the group of patients receiving oxygen, there was a statistically significant difference. So it was 4% mortality in the remdesivir group versus 12% in the placebo group. So based on those findings, the Australian National COVID-19 Clinical Evidence Task Force has provided a conditional recommendation that remdesivir should be considered for adults hospitalized with moderate to severe COVID-19 who do not require ventilation. And that's what that trial, the NIH trial, showed that this was the group that was most likely to benefit. And interesting, this is not the conclusion that the WHO has come to. And if you want further discussion around that, you can go to the Clinical Evidence Task Force website. So finally, immunomodulation. So we're now heading into the more severe end of illness, patients with acute respiratory distress syndrome, and look, how can we address inflammation in this setting? Again, going back to the recovery trial, this was really the first big study that showed a treatment that worked. So amongst all participants, about 6,000 participants, dexamethasone reduced the risk of dying by 17%. And this was particularly the case for patients on invasive mechanical ventilation, where the mortality reduced from 41% down to 29%, so a massive difference. Interestingly, for those patients who were not receiving oxygen, there was no evidence of benefit. And again, the kind of rate ratio actually trended towards usual care being better. But overall, dexamethasone is a clear winner, has clear benefit, and has now become the standard of care for the patient populations for which it should be used. And the recovery trial was then complemented by other randomized controlled trials and a meta-analysis from the WHO, which included, I think, 7 studies in total amongst critically ill patients, including the REMAP-CAP study, which is led by Australians. And this showed that all these studies favored steroids, and the overall effect using this fixed effect was a odds ratio of 0.66. So, 2/3 mortality rate if patients received steroids. So in conclusion, we should be using steroids for those who are requiring oxygen and hospitalized with COVID. I think remdesivir is reasonable to use that for those who are requiring oxygen, but not those in critical care. So once a patient's hit the stage of being ventilated in the intensive care unit, there's no benefit for remdesivir. Other antivirals are of no benefit, unfortunately. Maybe we could use monoclonal antibodies very early on. There's some other things on the horizon. So there's been press releases about tocilizumab, colchicine and anticoagulation. Keep your eye out for those. My final slide really is to, again, sing the praises of those who've been leading trials like Recovery and REMAP-CAP. So Martin Landre is the lead investigator for Recovery. These are the number of patients who've been recruited into this randomized controlled trial as of the 2nd of February, 34,000, I think it's 35,000 now. But basically every 2 or 3 days, they've been recruiting 1,000 new patients into this study. And this has really generated all this evidence that I've talked about today that helps to drive how we treat patients. They are still testing at the moment aspirin, colchicine, baricitinib, Regeneron monoclonals, and tocilizumab. And the REMAP-CAP is focused on critically ill patients, again, led by Australia and New Zealand. And they have also randomized about 10,000 or 5,000 patients with COVID into 10,000 different kinds of treatments. So it's a platform trial that assesses multiple things in parallel. So it's really the kind of amazing work that these studies have done, as I've said, that has led to where we are now, and knowing not just what works, but as importantly, I think, what doesn't work and what we shouldn't be using. Some people are saying that the recovery may well win a Nobel Prize in time to come. Thank you very much. I'll stop there.
Steve, thank you. That was masterful also. A very difficult area to summarise, and I think you've made it very clear for us. If you could stay online, and if Paul can join us, and then I think we've also got Dominic Dwyer coming in from Wuhan, maybe not. See, Dominic, welcome. For those who don't know Dominic is a virologist and infectious diseases specialist. He works at Westmead Hospital and New South Wales Health Pathology. He's currently in Wuhan as part of a WHO expert group looking at the early origins of COVID-19. We just to let people know, we might go a little bit over time. So if you don't mind, we'll definitely finish at 9:40. And Dominic's going to give a quick little summary of what he's allowed to tell us about what he's been doing and whether he's been to the market today or not. And then we'll open up for questions.
All right. Thanks very much, Lou, and hello to everyone. I'm sorry we're not meeting like we usually do down in Melbourne over some food and drink. So look, I don't know how you want to do this, Lou, but look, I've been here, I think, now 4 weeks, 2 weeks in hard quarantine in Wuhan, and then another 2 weeks, which in a sort of light quarantine. I've forgotten exactly. I think I've now had 7 swabs, 4 blood tests, and I'm getting my temperature measured about 4 times a day. It's just unbelievable. So by light quarantine, I mean that we've actually been given a sort of run of the wing of a hotel here in Wuhan, and we have to stay. We're essentially in a bubble, like a footy team or cricket team, basically. So we can all meet, and the local public health authorities have enabled us to go and visit sites that we want to visit and meet people that we'd like to meet. And then the rest of it is really very intense face-to-face discussions with Chinese colleagues. It's really quite remarkable. I'm trying to think what it's really like. It's a bit like doing a Viva for about 10 hours a day. You know, you drop your guard and it's all over. So it's actually.
And Dominic, how much of that is in English and how much of it's translated?
So it's all translated, either simultaneously or you know, one after the other sort of thing, which is an interesting exercise because we do have some Chinese people from the WHO office in China with us, and translators and the like. But it's interesting that, of course, the Chinese, quite a lot of them speak perfect English, but they do speak in Chinese. Fair enough too. I mean, we're the visitors, but it is interesting, and one has to be particularly careful then about people understanding what you might be saying just to a colleague. We visited all the high spots of Wuhan, the Henan market, of course, which is now closed, the Wuhan Institute of Virology, the various other public health labs, including the ones around 200 metres away from the market. You know, they've all been extremely interesting. It has also met people who've had the disease. So we've met who the Chinese regard as patient zero, and talked to him. And they sort of asked me to take a history from him in front of about 80 people. It's very intimidating. But we've also been to, for example, one of the communities here. Everybody pretty well lives in 50-story towers, but to talk to the impact there. And I did chat to one person who's actually an orthopedic surgeon, whose wife is an internal medicine physician. She died during the early stages of the pandemic, infected at work. He's sort of obviously left alone with a 2-year-old son. So it's actually quite interesting because you can spend a lot of time talking about numbers and hypotheses and figures and so on. But at the end of the day, of course, as everybody in this audience would know, this is a disease that affects individual people with you know, potentially catastrophic impacts, both physical and psychological. So it was quite instructive to do that. Part of the issue is dealing with a lot of the hypotheses, both sensible and ridiculous, and trying to work through those as far as one can with some of them. You know, we've been doing, well, the people here are in 3 groups. There's a group of animal experts, particularly in the area of bat sort of virology. And then there's a group who have experience in sort of, you know, clinical epidemiology and clinical virology and laboratory medicine. And then there's others that have expertise in some of the kind of public health aspects of outbreak evolution and so on. So we have been, well, you know, it's you know, clearly a diplomatic dance, a lot of this. There are reviews held on you know, the Chinese have views about origins and so on. And, you know, the difficult issue is trying to talk to people and put in the kind of politics aside. Obviously, it's heavily scrutinized. I mean, we're sitting opposite about 40 or 50 people at any given time. You know, obviously some of these are minders and so on. So it's really quite interesting. But when we do get down to the smaller group discussions, like you would expect, when you start talking to your own peer group and scientists and doctors and so on, you're much more likely to get a sort of feeling as to what's going on. It was very interesting going to the Henan market, the wet market, where this all exploded. And, you know, you go there and it's been closed for a year, so it's obviously different, but it's an absolute rabbit warren of a place. And really, sort of semi-open drainage and lousy ventilation, very crowded, and so on. And circumstantial evidence of the illegal presentation of wildlife products and animal products and so on. There's clearly, if you'll excuse the pun, an appetite to eat anything. And the role of that is really very interesting. There's a lot of discussion about the cold chain mode of transmission, and you may remember a very big outbreak in a market, is in Fadi market in Beijing, which really does look like it was related to cold chain transmission. In other words, frozen goods that were contaminated. And there have been a couple of other examples of that. I even heard Australian salmon being banned for it, which, that's okay in some of those scenarios. But in the original outbreak from the market, that probably doesn't really make sense, because when we've been looking, and we've actually been doing, line list examinations of the first cases in December 2019, only about, not even half of the people had a contact with that market. There was obviously transmission before and in the market. It's been, it's very hard to determine whether the market is a source. I think it's certainly an amplifying event. We all know that. But where the source is obviously really difficult. There are bats in a whole lot of countries in Asia and Southeast Asia and Africa and so on that carry coronaviruses of one form or another. But the distance between even the most closely linked ones and SARS-CoV-2 is, reasonable. I mean, if you sort of do a time to, ancestor time sort of analysis of sequences and so on, it really does put it back to kind of late November. So, how it kind of then unfolded a few weeks later in the market is really speculative. So anyway, and then so there's a lot of very kind of intense discussion and writing and so on, and it's obviously complicated and difficult. So happy to answer any questions if that's at all useful.
Yeah, Dominic, thank you. So there's a question that I think can be directed to all three of you, and it relates to the role of antibodies to COVID as measuring responses to vaccine, and potentially, I guess, to whether or not someone who's already been infected, needs to have the vaccine. Does anyone want to comment on? Well, we know the relationship in influenza, I guess, and that it's certainly not direct, and there are other factors. Is the same true for COVID?
I would say yes, basically. I mean, when we've measured the surrogates of immunogenicity in the early phase studies, they do seem to be loosely correlated with efficacious vaccines when we look at the clinical endpoints in the later phase studies, but it's certainly not linear. So, obviously the immunity has to take into account the antibody levels as well as the cellular component, and it seems a mixed response is probably ideal here. So it doesn't correlate exactly, but it's a useful surrogate, at least in the early phase studies. And I think the next part of that was about should the vaccine recipients be monitored, and certainly basically everyone who receives the vaccines will be monitored for adverse effects, and they'll obviously be reported through the appropriate channels. But I would like to see some more large-scale monitoring of responses and clinical outcomes. That's obviously a challenging thing to do, but I think we should try and do as much of that as we can.
Any other thoughts about antibodies?
Oh, yeah, look, no, Paul's outlined it well. I mean, I think antibody kinetics and so on in this in general are still sort of unknown. I mean, obviously people make a good antibody response after natural infection with IgG and IgM sort of dropping off pretty quick, IgM and IgA dropping off pretty quickly. The IgG persistence, I think, is problematic. I mean, we're really still only, at least in Australia, getting out to a year now, and people have been infected. So the long-term persistence of antibody, I think, is still uncertain. So then what that means for vaccine administration and so on. I mean, obviously in a country like Australia, it wouldn't be any desire, I wouldn't have thought, to do vaccine antibody testing or anything like that. But post-vaccine antibody testing and so on, I don't know, to tell you the truth. And again, I think we'll probably be hearing that data from overseas before we get it in Australia, I would imagine, given the way the disease circulation and the vaccine rollouts are happening. But I don't know that I have any super bright opinions on that.
Thank you. There's another question online related to sort of the logistics of the rollout of the vaccine, and at the same time trying to ensure that influenza is also covered. So given that there was virtually no influenza last year, is that a concern?
It's a tricky question, isn't it? I mean, I would think we certainly can't afford to take our foot off the pedal with the flu vaccine this year, especially if good coverage of the COVID vaccine facilitates at least some movement in the population. Again, obviously, we're not going to have widespread international travel, but if people start interacting more, we're going to see more cases. And I guess we're seeing that. I don't know if every state is seeing the same. We're seeing that with RSV, certainly up here in Queensland at the moment. So I think we do need to keep a focus on the flu vaccine, and it is going to be a challenge because it's essentially going to coincide with when our COVID vaccine is being rolled out, at least in that sort of 1A and 1B group. So, I think that makes 3 injections instead of 1, given the Pfizer vaccine is 2. So we're all going to be really busy, but I think we need to pay attention to the flu vaccine as well. And, not at the cost of the COVID vaccine, of course, but to try and do that as well.
Paul, there was an interesting question in the chat that you may have already answered, but it related to, can you swap vaccines halfway through? Can you have someone else's second dose?
Look, I think that the simple thing to say at this stage is we need to use the vaccines as intended and approved. And, the concept of heterologous boosting, so giving one and then giving a different one next, is certainly one that a lot of people are talking about, and it looks like it might have some really interesting properties. But, again, we just haven't done those studies to prove that yet. So my simple advice would be to have it as intended. I guess the challenge there is with AstraZeneca is we're not sure what that'll look like yet. It'll probably be a fairly big gap between doses with their new data. But I wouldn't be swapping one for the other outside of very exceptional circumstances.
Actually, just on the influenza, Steve?
I just wanted to ask Paul, will there be any concerns with co-administering the COVID vaccines and the influenza vaccine? You can imagine coming in and getting both if that was possible, but are there any theoretical concerns?
Yeah, I wouldn't do both at this stage. I guess we have to see what it looks like, and it'll depend a little bit on some timing and things, but I would just hate to see a misplaced allocation of an adverse event due to one versus the other. And so I would think a gap would be wise, and how feasible that'll be will depend on all sorts of logistical challenges. But I think the minimum recommendation is a week, but I'd perhaps recommend even 2, just to making sure it's really obvious.
Dominic, there's a question that from Alison Martin that you might be able to answer, suggesting that influenza figures might have been down because of reduced testing last year. Is that true, or was that true in New South Wales, for example?
Well, no, it certainly wasn't the case in New South Wales. So our system in the public sector anyway is that we have the rapid flu testing with the gene expert assay across 30 odd labs, or 35 labs, I think, in New South Wales. And the use of that was actually very high last year. It's just that the proportion of positives was negligible. We also, and it varied a bit from laboratory to laboratory, but even during, the height of COVID testing, we were still doing a lot of respiratory multiplex PCRs, and we're particularly focusing on, healthcare workers and that sort of stuff as well. And again, the prevalence of flu was absolutely negligible. There was plenty of rhinovirus. There was quite a bit of RSV and the occasional, adeno and so on mixed in, but no flu. It's really quite remarkable. And I don't have a good reason for it, other than I'm sure the travel or the stopping of travel into Australia has probably made a really significant difference. I mean, you can almost argue that influenza is genetically extinct in Australia, for what that's worth.
Maybe the viruses have done a deal.
Maybe
alternate years. But Steve, the response to steroids in COVID infection, should we be thinking about using steroids in other respiratory viral severe? So, for example, cytokine storm in influenza?
Certainly raises the question now, doesn't it, Lou? I guess we've never, no, it has been studied in other respiratory illnesses, but not to the same numbers to have such confidence in whether it works or not. And I think this idea of anti-inflammatories and steroids in particular for respiratory infections, we should be testing in a similar way to what's happening in recovery. We should be doing this for our community-acquired pneumonias, where we know much of it may well be related to an inflammatory cascade, particularly towards that more severe. And so, yes, but I think at this stage within a clinical trial, we should be doing those trials.
Could I just finish by asking each of you to give a sort of a one-word or one-sentence answer to what you think is going to be the biggest hurdle to achieving high vaccination rates in Australia? I'll start with you, Paul.
For COVID?
For COVID, correct. Paul?
Look, obviously we have logistical challenges and there's going to be issues with supply and all of those sort of things. But, I think uptake is going to be a challenge. There's still a lot of people saying, I don't want to be the first. And, it's hard to convince those people that they're going to be the 120 millionth person, which is the case now. So, I think it's been great to see the federal government try and address that. And I think it's going to be up to everybody. And we're going to need all hands on deck and a truly multidisciplinary approach. And, I think everyone that's come to this meeting is clearly going to be part of that. So, yeah, I think it's going to be that uptake issue.
Okay. Steve?
Look, I must say, I agree, but I'll just throw in there, we still don't know some of those key questions about duration of immunity and also impact of virus variation. So, if there is this immune pressure, will the virus mutate in a way that has some form of escape from our kind of antibody response?
Dominic?
Yeah, I think people have covered it, but I'd go for logistics, vaccine hesitancy and outright anti-vaccination stuff, and then, the long-term immunity from vaccination would be the case, yeah.
Thank you. Look, I'll close the session now. I want to thank all the speakers for this session and for the previous session. I'd like to thank the people behind the scenes who have allowed this to go forward. It's much more difficult than having a room full of people. Thanks, everyone.
Thanks very much, Lou.